Welcome to the first installment of the PacVec Fall Seminar Series. Today we have our training grantees, Gale Disler, who recently completed her master's degree in public health from the University of Alaska Anchorage. We have Renate Schlaht, a Doctor of Veterinary Medicine student from the University of Alaska Fairbanks. And we have Erin Taylor Kelly, a PhD candidate in vector biology from the University of California, Davis, present on their PacVec research today. With that, I will pass it on to Gale and Renate.
Hi, everyone. My name is Gale, and I will be presenting today with Renate. First, we want to say thank you to PacVec for inviting us to their fall seminar series, and also thank you to our mentor, Dr. Micah Hahn, for helping us with this project. We'll be discussing our project, focusing on understanding the role of veterinary personnel and pet travel in regards to tick and tick-borne disease in Alaska. To start us off, a little bit of background about ticks here in the state. So ticks, recognized as one of the main disease vectors for humans, domestic animals, and wildlife, has highlighted the concern regarding its increase in geographical range and the expansion and through the mobility of pets and the ability of ticks to find niches in their new environments, specifically in circumpolar regions. And while Alaska has historically been outside of the range for many of the medically important non-native tick species, recent research suggests that that may no longer be the case. A 2020 study established a baseline for tick surveillance here in Alaska. This study was a collaboration with state agencies, including the Office of the State Veterinarian, who have also been so kind to help us and work with us on this project. Through the study, it established a baseline using tick records that dated back to the early and mid 20th century, and found that there is an occurrence of 6 different tick species found in Alaska. These species were identified as having a historical presence and were noted as being native to the state. And as of 2019, 8 non-native tick species have been found in the state, though most of these found ticks were related to people or pets with a recent history of travel outside of Alaska, though not all occurrences of found non-native ticks had some form of reported recent travel. Looking over the decade between 2010 and 2019, we can see that tick records of non-native ticks has increased over the past decade. However, it should be noted that the significant increase in 2019 is probably likely related to the outreach efforts from both the study and the development of the Alaska Submitted Tick program, which is operated by the Office of the State Veterinarian and collects and categorizes ticks found in the state. Friendly reminder to anyone that's tuning in from Alaska, as well as anyone that is considering traveling up here, if you find a tick, please consider submitting it and turning it in to that program.
So ticks are obviously really important in pathogen transmission, and we're specifically concerned about it in our animal health, though there are a number of other reasons ticks are significant. So they obviously have significant effects to our pet health if we are having dogs run around the woods and there are non-native ticks up here with new pathogens. We're also keeping an eye on ticks in relation to our wildlife species because subsistence hunting is so important in Alaska. So there's a lot of surveillance efforts for Dermatha pictus, the winter tick, which could obviously have huge implications on our moose populations and would then affect the number of people who use moose for meat and materials. This is also significant for some of the small mammal species that other ticks can parasitize and are also used for meat and materials by a number of native communities in the area. They're also an issue for zoonotic disease, obviously, because the majority of pathogens that ticks carry, and that would be an issue for our domestic animals and our pets, would also be an issue to humans. And our pets are so close to humans that they are a huge reason that tick-borne disease gets passed. So this is that same list of non-native ticks that have been reported in Alaska. In parentheses are the reported hosts as found through the Submitted Tick program. And then underneath are the pathogens, some of the pathogens of public health interest that can be carried by these ticks. And as you can see, most of these non-native ticks are found very often on domestic animals and humans. And they carry a wide range of pathogens. A lot of these pathogens aren't known to be carried or able to be carried by most of our native ticks, so there are a few exceptions to that. So you can see why having some of these species make their way up to Alaska and establish in Alaska is so significant to our population that would be naive to most of these pathogens. So we set up this study to specifically look at ticks and tick-borne disease in the pet population using veterinary clinics because they have access to so many pets, and veterinarians, we believe, are one of the big educational sources for people in Alaska. So our survey or study had 3 parts. There's a veterinary personnel survey that looked at the knowledge, attitudes, and practices of veterinary personnel all over the state of Alaska. Each clinic participating also recruited clients and pets for a serosurvey and tick check, as well as an owner survey to get an idea of the knowledge, attitudes, and practices of owners. And then we did a lunch and learn education session as well because we wanted to have a significant education and outreach portion to the study, especially because of the low levels of knowledge and attitudes regarding ticks in Alaska, and we'll go into that a little bit more later. So we did recruitment via email. We called clinics because of our partnership with the Office of the State Vet. They sent out emails to their listserv as well as the Alaska Veterinary Medical Association listserv to help increase recruitment, show that they were aligned with us and that they were helping with our study, and to get word out to more vet clinics. And then because we're associated with UAF Vet Med program, which is one of the big recruiters for veterinarians back to Alaska, we had a number of personal contacts through that program that we were able to use to help recruit clinics. And all enrollment was done via a Qualtrics survey. So we had 10 clinics participate in some part of the study. We did have a clinic in nearly every public health sector in Alaska. So we had a really, really broad spread of clinics. A couple of those enrolled just for education, which was something we allowed because we were so focused on education in this project. We had 8 clinics enroll for sampling or for the project itself. And then due to some compliance issues, 1 clinic failed to actually sample for us, though they had completed the veterinary personnel survey. And 1 clinic sampled for us effectively, but didn't end up completing their veterinary personnel survey. So we had 7 clinics sampling pets and a different set of 7 clinics who had done the veterinary personnel survey. 6 of those overlapped. So we had, that resulted in 46 veterinary personnel surveys being filled out, 81 owner surveys, and 103 pets were sampled for ticks, as well as participated in the sero survey. And then due to some of those compliance issues that I talked about, not all of the owner surveys were able to be matched with a sero survey or pet sampling. So we had 59 of those that were able to be paired. Due to these differences in data, we analyzed each of these parts separately, as well as any of the paired data when we could.
For the veterinary personnel survey, our goal was to assess the knowledge, attitudes, and practices of veterinary personnel in Alaska with regard to ticks and tick-borne disease and the respective protective actions. The survey was available online through Qualtrics. It was any employee of the clinic that registered for this portion of the project could participate and take the survey. This survey was a single survey that featured 29 closed multiple choice questions and statements, including questions that were specific to veterinarians and only open to those veterinarians. The topics ranged from tick-borne disease awareness, local tick surveillance, client education, differential diagnosis, and preventative and protective actions. Upon completing the survey, clients were provided a link to the Alaska Submitted Tick Program's website through which they could learn more about ticks in Alaska, as well as order outreach materials or learn how to submit found ticks. To briefly touch on a few of the questions featured in each section, we asked veterinarians we asked participants whether or not they knew of the local tick populations in the state, and about half of participants and all of veterinarians indicated that they did. We also asked them about the Alaska Submitted Tick program, and very similarly, a little over half of participants and just over 90% of veterinarians indicated that they knew. Because Lyme disease is a concern for both the public health and the veterinary community, we did ask a few questions about it, including one about how long it takes for the pathogen to be transmitted via tick bite. And a little less than a quarter of participants and just over half of veterinarians indicated that they knew. We also asked questions about client education. And we've learned that when asking whether or not ticks are a concern for cats and dogs living in Alaska, about a little over 50% of participants said yes, that they felt that there was a concern here in the state. And when asking about client education, the overwhelming majority of participants felt that veterinary clinics were an important source of information for pet owners. And a little over half believe that they themselves play an important role in tick prevention and education at their work. However, in the practices, about 2/3 of participants reported that they rarely or never provide clients with brochures, handouts, and other outreach materials pertaining to ticks and tick-borne disease. And less than 20% teach clients how to check their pets for ticks. And none of the participants reported frequently doing so. We also asked questions regarding tick prevention, and when asking veterinarians if they felt that dogs and cats living in Alaska would benefit from tick prevention for some or all of the year, about 1/3 responded positively that they did feel like it would benefit those pets, and slightly more at just over 45% noted that they do recommend tick prevention for dogs and cats that spend time outdoors in Alaska. And because out-of-state travel has been linked to so many occurrences of non-native ticks found in the state, we did ask if they recommend tick prevention to dogs and cats traveling outside of Alaska. And all of the veterinarians participating in the survey responded that they do feel that it is important for dogs and cats traveling out of state. And again, all of the veterinarians also recommend tick prevention for dogs and cats traveling out of state.
So for the Zero Survey and Owner Survey, there were a couple goals for that part of the study, both a similar trying to understand what pet owners know about tick and tick-borne disease in Alaska and what they're doing about it, as well as trying to quantify how many ticks and what level of tick-borne disease is in our dog and cat population, because that's not something that we know about our pet population in Alaska. So we used 40x plus SNAP test, which is a really simple antigen test made by IDEXX that most veterinary clinics are familiar with. It does have pretty good sensitivity and specificity, and detects 2 species of Ehrlichia, 2 species of Anaplasma, Lyme disease, and then heartworm, which obviously isn't a tick-borne pathogen, but actually proved to be a really good way to recruit clients into the study because heartworm is tested for more than tick disease. And clinics found that they were able to recruit clients into the study with a free heartworm test if they enrolled in the study and filled out the survey. So we chose this test not necessarily because it was the best, but because it would be the most accessible for vet clinics to use. Most vet clinics are familiar with it. Most vet clinics either already use it or use another brand's version of a similar test. And it's also something that can be done in rural Alaska. So because we did have a number of clinics enrolled from rural Alaska where sending out blood isn't always really feasible or practical, they were able to do the SNAP test. It takes like 8 minutes and 3 drops of blood. So it's really easy, both on the animals as well as on the veterinarians. We were trying to add as little to the veterinarians' workload as possible. So what we found is we had 103 participants. There were 102 dogs and only 1 cat enrolled. Those tests are not labeled for cats, but able to be used in cats. There's a little bit of a discrepancy with that, but we told vet clinics that they could enroll cats if they wanted to, because we do think they're important and because we can get some data from that. As you can see, there was a low level of tick-borne disease in the population, and we got 1 anaplasmosis case and 1 Lyme case. The Lyme was linked with recent travel history, and I don't think it would have been tested if we weren't enrolled in the study. So we did catch a case of Lyme that was coming back into the state. We did not find any ticks while actively sampling, which is interesting, and we did sample for almost a full calendar year. We started in fall of 2020 and went through the summer of 2021. So we did end up getting all seasons in there. Some interesting things that came out of the owner survey, especially in relation to the veterinary personnel survey. If you remember Gale's statistics from a minute ago, is only a quarter of owners reported that they knew that we have ticks native to Alaska. And 50% of people thought that ticks were not capable of overwintering in Alaska, which is something we hear really often when discussing ticks in the state, that we don't have, we hear it with most ectoparasites in the state, honestly, that they think they can't live there because the winters are cold, which is obviously not true, and something that is really interesting that it's so prevalent in the owner population. Under 50% of people scored 4 out of 4 on some reasons that were presented to them that ticks might be a potential problem for people and pets, including irritation at the bite site, potential paralysis, and transmission of pathogens. But there were only 8% of people who believed that ticks were not a problem at all for dogs and people, which is encouraging. We also asked about attitudes and whether owners believed that ticks were concerning, and also about whether they believed ticks, that their concern for ticks would change in the next 5 years. And that gave us an interesting perspective, because only 30% of people reported that they were concerned, either at least somewhat or very concerned about ticks in Alaska right now. But over 50% of people did say that they would become more concerned about ticks in Alaska in the next 5 years. So that's showing that there is at least some idea that this is a changing issue in Alaska, either due to climate change, increased travel, etc. And then on prevention, only 13% of people reported that their pets were currently on prevention for ticks. That includes any sort of topical or oral chemical prevention. So it's not something that's super common in the state right now. And if you remember in Gale's statistics, 100% of veterinarians were recommending pets traveling out of state beyond prevention, but only 50% of people reported that if their pet left the state, they were on prevention. So that's an interesting discrepancy between what veterinarians are recommending and what clients are actually doing. So the lunch and learn is our educational session. We thought education would be really important for this project. And a lunch and learn, if you're not familiar with it, is something that's pretty common in veterinary clinics. It's a pretty common educational means in veterinary clinics where a drug company or some educational party will buy a clinic lunch and present, usually during the lunch hour, which is why it's called a lunch and learn. We wanted to do it as an incentive for sampling and as a thank you for clinics who effectively sampled for us, but as you said, the education portion was open to clinics that didn't sample as well. They just didn't receive compensation. So compensation was given via e-gift cards, and clinics had an option for a live presentation or a recorded video. So we recorded and listed and published on YouTube an educational video that went over kind of everything we went over in the background here, what the sampling is for ticks, knowledge about the Submit a Tick program, what ticks are native to the state and potential pathogens that they might be concerned about, as well as how we think that that's changing. And again, we did have a couple clinics enrolled just for the educational portion, which was encouraging because the clinics do care about these issues in the state and are interested in learning more about it. These clinics were also given a number of publications that we had used in our research, as well as things like the One Health Zoonotic Disease Prioritization Report that was recently published in Alaska, and any additional reading that they might be, for anyone who might be curious to learn more.
While the veterinary personnel survey and the client survey did not feature the same questions, they did include similar themes. When asking participants whether or not there were local tick populations in the state, we saw that about 100% of veterinarians and half of veterinary personnel were aware of those local populations, but less than a quarter of owners were. This could be highlighting potentially a knowledge gap within the different populations. We also, when asking questions about concern for dogs and cats in Alaska, about half of veterinary personnel felt that ticks were a concern, while about a third of owners did. A few of the public health takeaways that we gleaned from the different aspects of this project was the low levels of tick-borne disease in the population, as well as the fact that there were no ticks found actively feeding during the survey, though it should be noted that clinics did contact us letting us know that they found ticks outside of the survey period, so both before and after, but just not during. So we didn't get any of those for ours. Lastly, that there are discrepancies between the owner and the veterinary knowledge, attitudes, and practices frequencies. Like before, this could be indicating a education gap and could be a great area to focus future outreach on.
So as you mentioned, there were some challenges with this survey. A lot of them are related to COVID-19. A lot of issues with clinic and owner compliance, clinics not being super aware of what they needed to do, despite us reaching out repeatedly asking if they had any questions, and sending a lot of informational materials. There were holes in our recruitment. So despite the number of avenues we tried to recruit vet clinics through, we had no vet clinics in Anchorage enroll, and that's obviously the most populated area in Alaska. We don't know if that's due to how busy vet clinics are right now, or the fact that more vet clinics in Anchorage are corporate owned, which might limit their ability to enroll in things like studies. Again, similar to ticks, we know that not all seropositive animals were captured by the sero survey. That clinics reached out to me and said, oh, we had a clinic, or we had a positive case either right before sampling or right after sampling or during sampling, but they had used their in-house test or send out test or not our test for whatever variety of reasons. So we're looking at ways to capture that data as well. Because even though we were actively sampling, we definitely missed a number of cases. And then COVID-19 obviously is the bane of almost all research right now. It delayed our sampling. And then if you've brought your vet to the clinic, or if you brought your pet to a vet clinic right now, you know how busy they are. So many more people got pets, there's staffing shortages, vet clinics are really overloaded. And so many are still in curbside care, which this study was designed really around veterinary clinics and how veterinary clinics normally operate. And then when COVID-19 hit, we tried to adapt it. So things like the owner survey was designed to be completed in clinic with Wi-Fi, while the pet was being taken care of in the back, or with access to the veterinary personnel or the staff if people had questions or access issues. So that was not easily translated to curbside care, and I think that accounted for a lot of the compliance issues we had, and a lot of the owner surveys that we weren't able to link to sero surveys because we were doing that via participant ID, and that didn't always get translated to clients correctly because they were in their cars and not in the clinic. So that was a definite concern. We're still working on our analyses with the veterinary personnel survey. Gale's categorizing CAP scores with Bloom's cutoff points, and we're still working on associations between the demographic variables and the CAP results, as well as the associations between the owner survey questions and the veterinary personnel questions to get a broader idea of what all of those discrepancies in education might be. And we are working on writing up our manuscript for publication. So that will hopefully be worked on in the next few months. And then we just wanted to thank again PacVec and the Office of the State Vet was with us on almost every step of this. So they're the ones continuing the Meditech program, and that will still be going.
All right, thank you, Renata and Gale, for your wonderful presentation. Anybody have any questions for them? Please feel free to just unmute yourself and ask the question or use the chat box. All right, I do not see any questions, so we will move on to Taylor's presentation.
Great. Hi, my name is Taylor Kelly. I'm a PhD candidate at Aedes, and my advisor is Jeff Cardell. Yeah, thank you for inviting me to speak here today. I'll be talking about my PacVec work. So we are looking for biosignatures of pyrethroid resistance. Pyrethroids are important tools for adult vector suppression. As with many other chemical insecticides, they target insect ion channels and bind to the voltage-gated sodium channel, blocking the channel open, causing paralysis and eventually death. Unfortunately, resistance is a very serious problem. Insecticides confront multiple barriers on their way to their target site. Resistance mechanisms in mosquitoes can include thickening of the cuticle to reduce penetration, a great diversity of detoxification and excretion mechanisms, and target site modification. So modification of the voltage-gated sodium channel to interfere with binding. Typically, the way that we evaluate resistance in mosquitoes is through the CDC bottle bioassay procedure, where a 250 ml Wheaton bottle is coated with an insecticide and live insects are introduced into the bottle. They're monitored for knockdown time. We typically expect in susceptible populations 90+% of insects to knock down by a diagnostic period, and insects that survive past that diagnostic period are considered resistant. If populations have greater than 10% resistant insects, then we expect a control chemical to fail. So we do have some really important biomarkers of pyrethroid resistance already. Those include sodium channel mutations. These SNPs are present and also very prevalent in mosquitoes throughout California. In 2019, the California Department of Public Health published this extensive undertaking to surveil the prevalence of these mutations throughout Aedes aegypti populations in California. They found that 2 resistance mutations are basically fixed in mosquitoes throughout the Central Valley and very common throughout mosquito populations in Southern California. An important note is that mosquitoes in the Central Valley actually represent a different genetic cluster and genetic background than mosquitoes from the southern part of the state. In California, we find 5 voltage-gated sodium channel mutations, although only 3 of those are known to confer pyrethroid resistance. Those are in green, and all 3 are very near fixation, so prevalence of over 95% in the Central Valley. These biomarkers, though, do not perfectly predict resistance status of populations, particularly for some of the type 2 pyrethroids. So on the left, I have another figure from that paper from 2017, and they're reporting the frequency of resistance mutations to those 2 SNPs. And on the left are figures representing bottle assay performance of different populations run in 2018. So green is Rockefeller. It's a susceptible population that we use as a reference in bottle assays commonly. And the blue line represents a population derived from LA. The yellow lines represent populations derived from different cities in the Central Valley. So each of the yellow lines had mosquitoes that had all three resistance SNPs, but we can see there's variable performance in the bottle assay, and most of those populations would actually have been considered susceptible to deltamethrin. So our idea basically was to look for metabolites that could act as biomarkers for other mechanisms of resistance, and particularly for some of the diverse metabolic mechanisms, and point us towards pathways and also proteins that could be assayed potentially. Various specific detoxification mechanisms may have shared phenotypic consequences, and we expect that perhaps constitutive upregulation of mixed function oxidases may contribute to a more oxidizing environment in the body. Those are known to be involved in the resistance phenotype in Aedes aegypti. So metabolomic analysis can reveal differential demands on pathways, including those involved in mediating oxidative stress and regulating carbohydrate and lipid homeostasis. So that was our framework. And metabolomics allows us to study the insect physiology at a level which is very close to the biological phenotype. So we can actually see, what is going on in the insect system after they're presented with a challenge. So our strategy was to generate populations, so strains from these two different population clusters. So we developed F2 colonies from both the Central Valley and from the greater LA area, with the idea that by representing two different genetic clusters in the study, we can identify markers that are shared between multiple populations. So to collect our insects, basically we treated them to a modified bioassay procedure. So bottles were prepared as normal with deltamethrin, but they were exposed to the bottle environment only for 15 minutes and then released into white cages for monitoring. And basically we would watch them for knockdown, so the phenotype where they start to show symptoms of paralysis and inability to right themselves. And then they're collected and frozen on dry ice. So on the left is a chart showing our replicate numbers. So we also collected controls, so insects that were placed in bottles that were only treated with acetone. And then we determined insects were susceptible if they had shown symptoms of knockdown before 30 minutes. 30 minutes would be the diagnostic time in the typical CDC deltamethrin assay. And we chose a persistence past 70 minutes as our resistant group, because in previous studies, we had found that for mosquitoes that don't show kind of any symptoms of pyrethroid intoxication after 70 minutes, they go on to have a 24-hour survivalship rate of over 80%. So it's a good way of classifying also who's going to survive to the next day and beyond. So the greater LA mosquitoes are kind of in tones of green and blue, and they in general had kind of more susceptible individuals. So around 40% of the LA individuals fit into our susceptible category, whereas only 30% or so of the Central Valley populations fit this categorization. And in both populations, interestingly, about 30% fit into our resistance category. So we submit these samples to the West Coast Metabolomics Center on UC Davis campus. We look at assays for primary metabolites, lipids, and biogenic amines. The primary metabolite assay is certainly the best annotated, and we focus mostly on the known compounds. So in looking at kind of the clustering of our samples, principal component analysis is actually really useful for looking at metabolomics data sets. They're large data sets, and that is able to kind of compress some of the complexity to look at trends and groupings within our samples. In reading a PCA, it's very important to note that the first principal component indicates greater difference than the second. So kind of generally, we can see that like the LA group had really strong, clear separation of the susceptible and the resistant bugs. And we do see kind of a grouping of the resistant bugs, so yellow and dark blue from both groups. But there seems to be kind of a misclassification of a number of the susceptible bugs from the Central Valley. And this kind of pattern holds as we look at kind of graphs across the different principal components. It seems that about half, so 5 of the samples that were determined to be susceptible based on my criteria for the Central Valley have a metabolic profile that actually looks more like the resistant insects. This is interesting because we expected that group to be more resistant, and it would be interesting to further classify that group to kind of understand this. So just kind of a brief background of biomarker analysis. We do use MetaboAnalyst biomarker toolkit. This relies on some kind of basic machine learning math, specifically the generation of receiver operating curves. So I log transform my data, and some features look really different for the insects separated by class. The examples here are actually metabolites selected from the Central Valley group. It helps to look, I think, at instances where classification isn't perfect to kind of understand the graphs. So on the left is a rock curve for the Central Valley mosquitoes, and we're looking at the metabolite pseudouridine. So basically, you can kind of walk up and down the log transformed intensity of the metabolite and find a level that seems to best maximize the true positive rate and minimize the false positive rate, which this analysis determines to be 4.22. And then that is kind of a level that you can use as a classification standard. When you're looking at really large data sets, the chance that you find a random tightly correlated metabolite is pretty high. So it's often good to look at classification with multiple metabolites. And so that is represented with the figure farthest to the right. So the red line here shows the classification accuracy looking at 5 variables, including 10 improves the performance, but then adding more variables doesn't actually improve the performance of the classification that much for the Central Valley populations. However, the LA samples are very easily and reliably classified by a great number of features. So we found more than 20 metabolites just from the primary metabolite assay that basically work as perfect classifiers. So on the graph on the left, you can see the predictive accuracy of different numbers of features in splitting the samples into susceptible or resistant groups. And on the right is a table of features and the frequency at which they're selected in models. So metabolites that are selected more frequently tend to be particularly useful and accurate classifiers. So the figure on the left here shows the class probabilities of the different samples based on a model which only uses 5 metabolites. So the samples are very, very well segregated, very easily segregated. But we have more challenge in looking at the Central Valley mosquitoes. So we can see looking at just using 5 features in a model to classify these samples, the predictive accuracy is much lower. So it's improved to 85% with 10 metabolites, but it really depends on which biomarkers you're using in the model. That being said, we did find a number of shared features, which I think is really exciting. So the dynamics in one population seem to be shared with another. So looking at the primary metabolite data sets is much easier, I think, than looking at some of the other data sets, which are not as well characterized or annotated. So we took advantage of the Fine Lab at UC Davis. They made a chemical similarity enrichment toolkit. So we can look at different kind of cluster dynamics in our data. So on the Y is p-value, and then along the X is actually basically like the tree of different types of chemical groups. Groups that are together are kind of more chemically similar. So in this corner, we see dynamics with lipid metabolism. And that is basically the feature that I want to go ahead and start to detail first. So we see these kind of biosignatures of lysophosphatidyl-choline and lysophosphatidyl-ethanolamines. And that's really interesting because we know that LPCs are immune activators in animals, and they often show up in stress experiments. Some of them have been associated with antimicrobial properties, and dysregulation is associated with a number of human metabolic disorders. Phospholipase C, which actually liberates a fatty acid from phosphatidylcholines and phosphatidylethanolamines, may be constitutively overexpressed, and I think that would be really interesting to look at as a target as a synergist or something like that. In a previous study, we had compared Clovis mosquitoes, which are also a Central Valley population, to Rockefeller, which is our susceptible, lab reference colony. And it's interesting to see that some of the features that we pulled out in that experiment actually do seem to show up in this second experiment where we're doing an exposure and then segregating by phenotype. In particular, are these 2 LPEs and 2 LPCs. So we know that these can also activate different cell signaling cascades, and the overexpression of phospholipase A2 can be important to vascular and neurodegenerative diseases in animals. So it would be interesting to kind of better understand the relationship between these LPEs. Related is actually some dynamics that we've noticed with B vitamins as biomarkers in these samples. So vitamin B5 is an important cofactor in fatty acid and carbohydrate oxidation. Beta oxidation of fatty acids is very important in mediating pyrethroid toxicity. Vitamin B5 is also a precursor to coenzyme A. So we think it may be interesting to pursue why resistant insects are so much better able to kind of mobilize lipids to participate in these reactions. So understanding kind of the mobilization of these lipids would be interesting to understanding the actual mechanisms of metabolic resistance. Second, a marker that we identified in both studies is elevated levels of biotin. So biotin is B7, and it's an essential cofactor and coenzyme of a number of carboxylases. Similarly, it's involved in lipid and amino acid metabolism. But previously, biotin binding proteins have been proposed as insecticides and identified to have insecticidal activity in larvae and adult mosquitoes. So we hypothesized that potentially pyrethroid resistant bugs may be particularly susceptible to these compounds because they appear to be basically deficient compared to their susceptible counterparts. The last feature that I wanted to talk about today is pseudouridine and basically the epitranscriptome. So one of the strong classifiers we've also identified in both studies is pseudouridine. It's the most common modified nuclear base in RNA, and it can improve the binding affinity and stability of various RNA structures. So it is elevated in resistant insects following deltamethrin exposure. And it's known to be particularly important in ribosomal RNA and transfer RNA because it can help maintain their mRNA processing and translating abilities. So we think basically that this biomarker is indicating that the resistant bugs are stabilizing transcripts and kind of better able to mobilize machinery to deal with pyrethroid toxicity. Pseudouridine elevation is commonly identified in stress exposure experiments. So oxidative stress in mammals and yeast and following muscicide exposure in snails. It is interesting to see that it was slightly elevated in Clovis compared to Rockefeller mosquitoes, but it really does seem to show up following exposure. So while it's not necessarily a particularly interesting feature on its own, I think identifying mechanisms of regulation of pseudourination could be particularly interesting in better understanding metabolic mechanisms of resistance. So in conclusion, we do see good classification, but we see classification issues in the Central Valley population. We think that this could potentially be resolved by selectively generating genetically related susceptible and resistant mosquito lines. So taking the LA and Central Valley populations and selecting for susceptibility and resistance, and then basically reevaluating some of the things that we have found here. Large numbers of metabolites do effectively classify our susceptible and resistant mosquitoes following exposure. It'll be interesting to continue to try and find features that are more innate and don't require exposure. But these signatures are already pointing towards a better understanding of the mechanistic underpinnings of metabolic resistance. So our next steps will be to do some line selection and try to replicate our findings and also investigate tissue-specific kind of abundance patterns of some of the interesting metabolites that we found. And also to investigate the classification accuracy of these biomarkers eventually on wild samples. So I want to thank our lab, Jeff and Lindsay. We also got a lot of help, especially in mosquito collection from the Cornell lab. And I wanted to thank Susan Clue and Mir Bear Johnson for working with us to help us get mosquitoes, as well as PacVec for supporting this work, and the West Coast Metabolomics Center for all their help.
All right, thank you, Taylor, for your presentation. Anybody with questions, please feel free to unmute yourself or to send them through chat.
Taylor, I had a quick question about what to make of the LA versus Central Valley populations. And I know with KDR, it's been pretty fixed in the Central Valley. But then with the metabolic assays you're doing, you found a much more mixed signature seemingly, where you couldn't really classify them in the Central Valley, whereas LA looked clean, which is just the opposite of what you get with KDR. So do you think that means that Southern California resistance is more driven by metabolic mechanisms versus KDR in the Valley?
I think there are a couple lines of evidence to support that, including the second paper that was published by the CDPH, but also it was kind of something that we were able to look at when we looked at knockdown and controlled for genotype. So when we did that, we found that the greater LA mosquitoes had kind of on average just much longer knockdown times. So yeah, controlling for genotype, they seem to do better. So that would indicate, yeah, that they seem to also have stronger metabolic resistance. Yeah, the relationship between SNPs and resistance really depends on the insecticide, the specific, very specific chemical. So.
Yeah, and the way you're selecting those mosquitoes seems like, I mean, KDR would be part of that mix clearly, where you're holding them for 70 minutes, and it looks almost like the Central Valley population might be more cleanly KDR based and not metabolic at all.
Yeah, that could be. It would be interesting to think about the different pressures that LA mosquitoes might be kind of coming into contact with. But the KDR is a great biomarker on its own for permethrin resistance. But they don't always generalize very well past that.
Yeah, really nice work.
Slide about the elevated biotin in the susceptible populations. I guess if the elevated biotin was in the susceptible populations, doesn't that mean that the biotin binding proteins wouldn't be as valuable as insecticides for the resistant mosquitoes?
Well, so I'm not sure, but I think one of the things that we might be seeing is basically that the biotin is consumed at a higher rate, such that the resistant bugs are potentially biotin deficient. And in that framework, then applying the pressure of biotin binding proteins, they're going to see kind of a stronger reaction because they're going to be more susceptible to losing the biotin that they have available because it's already reduced.
Okay.
That was
That
how
makes
I was
sense.
thinking about it, but who knows? I don't know.
No, that seems reasonable. Okay, thank you.
Okay, thank you so much to Taylor and Renata and Gale for your presentation today. Thank you all for coming to our first seminar as well. The next one will be on Tuesday, October 12th at the same time from 12 to 1 p.m. Pacific time. So have a good day and bye-bye.